And the method, why the method is called ELISA, because the detector molecule that are the secondary antibodies are linked with the enzyme, and during the assay process, when a substrate specific for that enzyme is provided, it change the color, and then based on the color detection, this method is used for the qualitative or the quantitative method of quantifying or quantitating a particular pathogen. By means of the principle, there are two different types of the ELISA: indirect ELISA and the sandwich or the direct ELISA. As it is quite obvious from the name indirect, it means that this method of the ELISA, it detects basically the antibodies raised against that particular pathogen or antigen. While in the sandwich or the direct ELISA, it directly quantitates the pathogen or the antigen that is present if the person is infected and present in the serum. So in case of the indirect ELISA, the antigens, they are coated along the wall of the well. So the serum is taken or the blood sample is taken from the patient and poured inside this well. So if the patient is really infected with the SARS-CoV-2, then obviously it will have, the patient would have generated the antibodies. It will then attach with those antigens. So if the patient is really infected and the antigens are attached and the antibodies are attached with these particular specific antigens, then again a secondary antibodies linked with the enzyme is then poured inside this well or in the enzyme assay. So if the antibodies are attached, it will attach with these antibodies. It means that these antigens are anti-human antibodies. So if these antibodies are present, obviously these antibodies will attach. But if these antibodies are not present, so these antibodies has no affinity for these antigens. So obviously, during the washing step, these antibodies will be eluted. So if there are two cases, if the antibodies are present, the person is infected and the antibodies are present, these antibodies will attach with these antigens. So after the washing step, non-specific antibodies will be washed out. Then the secondary or the detector antibodies linked with the enzyme will be added. It will attach with these antibodies, and then the substrate will be added, and the substrate will definitely change the color. So upon change of the color, it will be detected that the person is infected and the person has raised the antibody specific for the pathogen. Now in case number 2, if the person is not infected with the SARS-CoV-2, so what will happen? The patient definitely would have not generated the antibodies. So the antibodies will not attach with these specific antigens. After washing, when the secondary antibodies will be added, as these antigens are specific only for the SARS-CoV-2 specific antibodies, it is not specific for these antigens. Of course, after the washing step, these secondary antibodies will also be eluted. So if these secondary bodies will be eluted, the enzyme will also be eluted. So when in the last step the substrate will be added, it will not change the color. So if the color is not changed, it indicates that the person is not infected with the SARS-CoV-2. On the other hand, in the direct ELISA, the wall of the well is coated with the antibody specific for the pathogen or the antigen. So once the patient's serum is taken and added inside the well, the antigen will bind with the antibody specific for those antigen. And then the secondary antibodies will be added for the detection purpose. So as the secondary antibodies would also attach with the same antigen but with the different epitope, and the pathogen will be sandwiched between the primary and the secondary antibody. That's why this direct ELISA method is also called as the sandwich ELISA. So there are two cases. For example, if the patient is really infected with the pathogen, the antigen will attach with the antibodies coated over the wall of the container. The secondary antibodies will also bind with its second epitope with the antigen. When the substrate is added, it will change the color, and upon change of the color, it will be the indicative of having the infection. Now, case number 2, if the person is not infected, obviously the serum will not contain the pathogen. So if the antigen or the pathogen is not present, the antigen will not attach with the antibodies. So upon adding the secondary antibodies, which is only specific for the antigen and not for these antibodies, so upon the washing step, these secondary antibodies will be washed out. So as the secondary antibodies will be washed out, it will also take away this enzyme linked with the secondary antibodies. So upon adding the substrate, it will not change the color because the enzyme is not present. So it will be the indicator of the opinion that the patient is not infected. The next method, which is the most popular one and the most accurate one, is the polymerase chain reaction, which we simply call as the PCR method. So we call this polymerase chain reaction because the polymerization process occurs in every step. For example, the 40 cycles of the polymerization or the replication happens, and then at every cycle of the replication, it doubles the quantity of the DNA. This method is basically requires these components: the template DNA, which in case of the SARS-CoV-2 would obviously be an RNA particle; the primer specific for detecting the target sample; the nucleotide as the raw material; the Taq polymerase enzyme, which will amplify the target gene; the buffer for the polymerase enzyme. So by mixing all these substances and putting them in the thermocycler, the primer during the denaturation step, basically there happens 3 steps during the polymerization process. Denaturation, which detaches or dissociates the double-stranded DNA from each other. Annealing, when the primer is attached with the target DNA, and extension, when this primer is elongated during the process of the replication. And all these 3 processes happens at the different temperature. For example, denaturation occurs at 95 degrees centigrade, annealing occurs at 55 degrees centigrade, and the extension of the polymerization, it occurs at 72 degrees centigrade. So as this method, the PCR method, particularly for the diagnostic purpose, it is totally based on the Taqman primer probe based method. So in that method, the primer basically recognizes the target sequence, while the fluorescent dye is attached with the probe. The probe basically on the one end, it contains the reporter dye, whose glow basically indicates the presence of the particular target DNA, while on the other hand, it has the quencher, which basically controls the fluorescence of this fluorescent dye. So as long as the probe is intact, the quencher, it controls the fluorescence of the reporter dye. So during the replication process, when the primer extension happens, the polymerase enzyme has the ability to degrade that DNA particle, that oligonucleotide particle which is present in its way. So once during the replication process or during the primer extension process, it degrades this probe which is attached with the target, and the target is basically present in between the 2 primers. So once it degrades this probe, this quencher molecule is detached from this reporter dye is detached from this quencher. So the quencher is no longer in close contact with the reporter dye. Once the reporter dye is detached and it comes apart from or at a distance from this quencher, it then releases the glow. So this amplification indicates that the glow will happen. So it means that if the viral genetic material is there, obviously the replication will happen. So once the replication will happen, obviously it will degrade the probe particle. So once the probe will be degraded, it will release the reporter dye, and the reporter dye will release the fluorescence. And upon detection of the fluorescence, will indicate the presence of the pathogen, presence of the genetic material of the pathogen. So this one is the whole the protocol of the diagnostic process. RT-PCR based method is workable for the nasopharyngeal or the oropharyngeal samples. So the sample is collected from the nose or from the buccal cavity. As the genetic material of the SARS-CoV-2 virus is composed of the RNA, so primarily it is subjected for the reverse transcription in order to get the cDNA particles, because the thermocycling condition works on the DNA basically. So after this reverse transcription process, the normal amplification process of the viral cDNA particle will happen. So if the sample really contains the virus, obviously during the reverse transcription process, this RNA particle will be converted to the cDNA. And when the cDNA particle is subjected for the polymerase chain reaction, the probe will definitely be attached with its target. And during the extension of the primer, the polymerase enzyme will be broken down this probe, and the reporter dye will be detached from the sequencer, and it will release the fluorescence. So upon release of the fluorescence, we will get the result. And the result basically depends on the CT value. CT value means cycle threshold value, that at which cycle the fluorescence is going to be detected. It depends upon the viral load. So if the viral load is high, there will be high content of the DNA. And if there will be high content of the DNA, we will get the detection early. But if there is low content of the RNA particle, there will be low cDNA. We will get the low value of the cDNA, and the CT, and we will detect the CT value, or we will detect the fluorescence in a very late stage. So early CT means that the patient is positive for the viral particle, and late CT, or there is no CT at all, indicate that the patient is negative for the viral particles. So summarizing the whole story, the molecular diagnostic testing is based on the nasopharyngeal samples, then the extraction of the DNA and the RNA, reverse transcription process, and detection of the fluorescence. While the serological test is based on the blood samples or the serum samples, and it is either the lateral flow immuno-based assay or the ELISA-based assay.